In this lecture, we'll introduce lipids. In particular, we'll be discussing various lipid classes, including fatty acids, triacylglycerides, some various other structural lipids, and we'll talk about membrane structure. In particular, we'll talk about how membranes are composed, what lipids membranes are frequently composed of. We'll talk about membrane fluidity, and we'll talk about some of the proteins that might be associated with membranes. We'll also talk about membrane trafficking, in particular getting into and out of cellular compartments, and we'll also talk about transmembrane signaling, in this case getting information across the membrane. Beginning with fatty acids, so these are our basic building blocks for lots of lipids. Here we see the fatty acid chain itself with a polar head group, a hydrocarbon tail, in this case a completely saturated hydrocarbon tail, and you notice that there's an alpha and omega, the alpha carbon being that carbon next to the acid group, but the whole thing referred to as the fatty acid. Let's see there, just in terms of nomenclature, here's a C18 fatty acid, known as stearic acid, because it's derived from steers originally. That's a solid at room temperature. Oleic acid, also C18, but here we see what the standard nomenclature for degrees of unsaturation in the fatty acid chain. So between carbon 9 and 10, there is a double bond. You can see that this introduces a kink in the structure there. We'll write that as 18,1 delta 9. And then we'll see some other examples wherein we have multiple unsaturated bonds, and then we'll slip those in after that delta as well. Interestingly, this simple difference introduces some very complicated behaviors, just in terms of the fluidity of the fats or the oils. Whether something is considered a fat or an oil really is going to depend on whether it's a solid or a liquid. Oleic acid is a liquid, stearic acid is a solid. They only differ by this one bond. How does that come about? Well, you can imagine that the stearic acid packs easily with other partners. You can rotate around all those bonds and it'll form a fairly compact structure. Whereas oleic acid, this cis double bond here makes this a more bulky structure, and we don't get rotation around that bond, so we occupy more space. Therefore, we're unable to pack as well, and we are a liquid at room temperature. If we look at some composition of fats from different sources, we're looking at olive oil here. That's a liquid at room temperature, and we see that it's mostly unsaturated composed of unsaturated fats. And as we move on to beef, which is a hard solid, we see that that's much more saturated fats, right? No double bonds. And we see that overall, we're looking at fatty acids. Most of the time, they're between maybe 16 and 18. The smaller, shorter chain fatty acids are less common overall. What else can I say about these? Typically, we'll be seeing in nature fatty acids that range from 4 to 36 carbons. Usually we're going to be talking about even numbers. Usually we're not going to see any branching in this chain, although that's not strictly true. And we'll frequently see certain amount of unsaturation. Again, this is the beginning of the story of how we control membrane structure. The idea is that by varying the composition of lipids, we can vary the properties of the supramolecular structure that is the membrane. Last thing I'll say here before I leave is that normally in biology we're going to encounter cis fatty acids. We're not going to talk about the nutritional issues with trans fatty acids, but you can imagine that those trans fatty acids are going to behave more like an unsaturated fat, right? They're going to be, even though you have that unsaturated bond, they're going to behave more like a saturated fat, in that they're more compact, they pack together more easily, they form a solid more easily, maybe they form a a solid structure in your blood vessels, for example, more easily. So they maybe have more of a tendency to form plaques. Um, However, we won't go too deeply into that. Just to say that with trans fats, the actual chemical mechanism of um, their dangerous or health dangers is really not that well understood. Instead, what we rely upon is as a statistical measure of how frequently we see a correlation between ingestion of trans fats and heart disease. Okay, so what do we do with those fatty acids? Well, we build them up into triacylglycerols. We store our lipids as triacylglycerol. So here's a glycerol. We've seen this structure already. We can esterify that at either any of these three positions. So a triacylglycerol has been esterified at 1, 2, or 3 positions, and we can have a simple triacylglycerol. Simple, that is to say, each position has the same fatty acyl group attached to it, or that we can have mixed. Then we'd have up to 3 different functional groups attached at each position. There are lipases, which we'll discuss only briefly, that liberate those fatty acids, and we'll see, however, that can be very important in terms of signaling. So right away, um, let me point out that the fats will serve a variety of functions, as is common in biology. We've seen this as well before too. Structural, right? These are we're talking about the molecules that form the lipid membranes of all of our cells, as well as nutritional. Triacylglycerols are a wonderful way to store energy for later oxidation, as well as signaling. So these molecules, the liberation of these fats, we'll see playing a role in intracellular signaling. Last thing I'll say about these guys is that it's you can store this away for your metabolism section of this course. Uh, Lipids are a better source of energy than carbohydrates in terms of uh, the the amount of energy that you can store. Um, they're more reduced than carbohydrates, for example. But the big key here is that they're nonpolar, and so they don't get hydrated. And so with carbohydrates, on a per mole basis, you're storing less energy because you have to pack a bunch of water around that as well. So by mass, you're storing less energy, whereas lipids, you can store that much more densely. Okay, so structural lipids, let's start out with this. Again, there's glycerol. We see that in the glycerophospholipids, we will attach 2 lipid chains to our glycerol moiety, and then we'll have some polar head group here. Now that's going to start with a phosphoryl group, but we'll frequently add something else to that. Myriad, that has a specific definition in Greek, but for all I mean is that we can form a variety of different substitutions. Frequently we'll see, though, that C1 is decorated with C18 or C16 fatty acids, and C2 is often slightly longer and perhaps unsaturated. So this is the core structure, phosphatidate, and is often further modified. As I said, that head group can also bear choline, serine, ethanolamine, inositol, glycerol, and these, again, as with the variation in the C1 and C2 modifications, these modifications alter the chemical characteristics of the head group, which will be very important, again, in determining the complexity of membrane structure. Okay, not only glycerol phospholipids are interesting to us, but another type of lipid known as the sphingolipid. Um, Now, I think your book describes this. I won't go into detail, but the name is kind of cute. Sphingolipids, referring to the fact that when first discovered, the actual biological utility of these lipids was poorly understood, so they were mysterious as the Sphinx. For us, we won't think of them as being particularly different. We see that we have a similar structure in that we have this glycerol-like um, backbone. We see that we've got a couple hydroxyls, we've got an amino group here, and then we have one long tail here. We can attach a fatty acid by a peptide bond or an amide bond, more properly right there, and we'll frequently see these in the vertebrate lipid component, frequently in neural tissue, so in the brain, for example. And that's all I'll say about these. We'll see more sphingolipids in class. Um, Something else to mention, of course, is that uh, as I mentioned earlier, we can place a variety of head groups here. Then this is not a phospho. Um, this is not a. You notice this is not a glycerol phospholipid. There's no phosphoryl group here, but we have modified that carbon, that third carbon with a carbohydrate now, and this could be an oligosaccharide as well. Um, Frequently, that's going to be galactose. So, in plant membranes at least, galactolipids are a common component. Um, Here again, this is a cerebroside. This is a sphingolipid again, and we will see that there's our amide linkage to at the C2 position for another fatty acid chain. There's our standard sphingolipid tail, and then we've attached it to a carbohydrate that can be simple or complicated. Now, this is a nice, maybe a little bit overwhelming uh, overview of the lipids. Um, Again, the triacylglycerols will refer to these as storage lipids. Right, they'll typically be found in adipocytes as storage as oily droplets. Um, and then we have a whole variety of polar lipids. That is to say, we've modified them with a polar head group. We've got the phospholipids, we've got glycolipids, and then there are other ether lipids that we're not going to talk about that are typically found in the archae. Just interesting to point out, though, that these are much more complicated in that there's two glycerols, and so we have this cyclical structure tying together our two fatty acid chains, and then we've got polar groups at each end. Very stable structures, I should say. But if we look closely here, let's go back to the phospholipids again. Glycerol, lipid, fatty acid, and then some phosphoryl group with a variety of potential other head groups, and then we see something similar for the sphingolipids, phosphoryl group, and then some other polar head group, and then in the glycolipids we've modified either sphingosine or that glycerol backbone with a mono or more complicated carbohydrate, or even a sulfuryl group. I'm going to only talk briefly about this, but the point is that, as I mentioned earlier, we'll leap ahead to the idea of signaling with lipids and then liberating lipids for transport across membranes. In the diet, for example, we'll use pancreatic lipases to convert triacylglycerols into the fatty acid chains for import, and then we'll tack them back together into triacylglycerides again on the other side of the membrane. Lipases are esterases, same catalytic triad type structure to hydrolyze that acylglycerol ester bond. We won't go into the mechanisms there. Um, let's just say that the fats that we digest in this way, we assemble into a complicated micellar structure. I mentioned micelle structures to you at the beginning of the term. We'll see a couple of examples here again, but just remember that a micelle is a structure that allows us to deal with both the polar end of these molecules and the nonpolar end. So we form a sort of a droplet in which the nonpolar tails are facing in, and we decorate the surface with more polar head groups. These micellar components enter the intestinal enterocyte either by simple diffusion across the membrane. So they just merge with the membrane and then there's all more active transport as well. Again, we're not going to go deeply into those details. But I should just say, as with sugars, when we ingest a, a disaccharide like sucrose, of course, we're not going to import it into our cells as a disaccharide. We break it down first and import, and then the same is true of these triacylglycerols in the diet. Different kinds of phospholipases. These are just general classes of lipases pointing out where we cleave. We'll liberate those fatty acid chains by using phospholipases type A. A couple of subtypes there, please note. We'll liberate the head group away from the phosphoryl group with the lipase D, and then lipase C will liberate that whole phosphoryl plus head group. So you can imagine here, not only are we talking about degrading a lipid perhaps for use as an energy source in fuel oxidation, but also for liberating signaling molecules. You can imagine, and we will see, the use of the head group as a signaling molecule and a use of the tail groups as signaling molecules in different contexts. Okay, let's think about how fats are transported. Again, a very cursory look at lipoproteins. There are a variety of different types of lipid organization structures. Um, these are found in the plasma, right? So we're referring to humans in this case. The simplest being the chylomicrons. These are very low density, right? Think about what before we get started, what does low density mean? Well, it means it's very high in fat content and maybe very low in relative protein content. So less dense than water, for example. As you're all aware, if you've every time you shake up your vinaigrette, the relative density of these oils. Um, Lots of triacylglycerides in the chylomicrons. They're um, yeah, usually carrying our dietary lipids again across the membrane and as we ingest our lipids. Very low density lipoproteins. These are typically composed of synthesized lipids and for distribution to the tissues. Um, Low density lipoproteins, um, so they're slightly higher density than the very low density lipoproteins. These are relatively high in cholesterol, and this would be our bad cholesterol, which I'm sure we're all familiar with that terminology. Low density lipoproteins are carrying cholesterol from the liver out to the tissues. And that's a bad thing because that could lead to the deposition of that cholesterol in the tissues, which we normally would associate with disorders such as various kinds of heart disease. The high density lipoproteins, on the other hand, are carrying the cholesterol back from the tissues to the liver for storage or degradation. So um, that's our good cholesterol. And it's very easy to remember. The high density lipoproteins are carrying the lipids back from the tissues for degradation. That's good. Delivery to the tissues is bad. Of course, however, we obviously need to deliver cholesterol and other lipids to the tissues. It's just a matter of how much. This cartoon down here is nice. I suppose we're looking at a low density lipoprotein here. Just to give you the sense of how these are composed, in blue we have this protein component. This is apoprotein B100, whatever that means. Doesn't really matter for us. But we note that the composition of proteins in these lipoproteins helps determine the distribution route or ultimate destination of these lipoproteins through mechanisms that we've already discussed, for example, glycosylation. And but in any case, let's look too at the surface of this structure, covered with polar lipids and maybe cholesterols too, sort of so forming this sort of relatively hydrophilic shell that hides all of our triglycerols and cholesterols in the core. So a water soluble shell to carry our water insoluble lipids into the tissues. Let's look at a few more lipids. The sterols. I haven't drawn a sterol here. If we switch out this carbonyl with a hydroxyl, this would be cholesterol. But just structurally, let's think about this. We've got four fused rings, A, B, C, and D, relatively rigid, not terribly planar, but more or less planar. We're usually going to have some polar head group and various modifications here at any of these different positions. Derived from isoprene, which is this structure, which is a different route for biosynthesis than the acetyl-CoA from which the fatty acids are derived. That's something, again, that we're going to not get into until second semester. Waxes, we won't spend on any time on these. These are, again, structural molecules. We see that they're um, uh, but mostly composed of long chains, right? With 14 here, 29 here, with this kind of complicated linkage. Um, Lots of high degree of unsaturation, but usually we'll see these as water repellents, for example, in insect tissues and in plant tissues. let's talk about lipids as signaling molecules. I alluded to this earlier. Here is a, we don't really care what lipid this is, but we note that it's a glycerol-based lipid. There's some head group that doesn't matter, and what we see that in terms of the fatty acids here is that one of these fatty acids, again, I mentioned that frequently in mammalian tissue, we'll find these these glycerophospholipids with a saturated chain at C1 and an unsaturated chain at C2, and indeed here we have a 20 carbon chain with 4 degrees of unsaturation. So this would be 24 delta 5 8 11 14. And this molecule is liberated by a phospholipase type A to reveal this eicosanoid. So this is a 20 carbon lipid known as arachidonic acid, and you see that it has this could form this can form this kind of interesting structure. But arachidonic acid itself is in control of a variety of cellular signals. Let's back up for a second just to remind us, though, that 20 carbons, this is reminding us that we use a Greek nomenclature frequently in the fatty acid. So ICOS telling us that that's a 20 carbon chain. We'll talk more about the Greek system in class. But in response to various kinds of stimuli, phospholipase A releases this arachidonic acid, and this molecule undergoes a variety of further modifications, resulting in the formation of prostaglandins, which are involved, and thromboxanes, which are both involved in inflammation. And so these are also related to a bunch of interesting drug molecules known as the NSAIDs, or nonsteroidal anti-inflammatories, that inhibit their signaling pathways, thereby attenuating inflammation responses. Those of you that take NSAIDs, you're probably familiar with that. As well, though, the arachidonic acids give rise to the leukotrienes, which are involved in muscle contraction. And there are also drugs that are in arachidonic acid analogs, or leukotriene analogs, that inhibit muscle contraction. Particularly, these drugs are useful in treating asthma. There we were looking at fatty acids as signaling molecules. Let's look at the sterols as signaling molecules. Here again is another wonderful Goodsell cartoon. Let's see, as the lipid molecules as acting as transcriptional activators in this case. So here is the estrogen receptor, and this is the hormone binding domain. You see in purple the estradiol ligand. This is estradiol here. This is a sex hormone. There's part of the structure that we're not seeing, which is controlling the interaction of the whole structure with RNA polymerase, which of course reads the DNA and makes our message. And then this would be the DNA element, referred to formally as the hormone response element, clearly interacting intimately with the DNA. But the idea, however, and we won't go into this pathway in great depth, is that the presence of the hormone causes the receptor to dimerize, migrate into the nucleus, and interact with the hormone response element, activating or repressing transcription of a whole variety of target genes. Prednisone is another signaling molecule that's more involved in inflammation. Okay, well, I have threw in a slide here about fat-soluble vitamins. Again, this will be sort of looking ahead for a lot of you that go on to take 461 and to discuss metabolism. But just suffice it to say that we won't talk about vitamins more than in this lecture in 460. However, it's clear that there are probably a set of water-soluble vitamins that we're not going to discuss, but those are the B vitamins, and then the class of fat-soluble vitamins that are derived both from the sterols and from, this is also actually an isoprene derived, so both of these are derived from isoprene as a source. So here we have vitamin D involved in regulation of calcium deposition and uptake. And here we're looking at the conversion of beta carotene into vitamin A or retinol. Um, this is an interesting molecule. Um, we're going to see it again in a few slides. Um, but bottom line is that the cis-trans isomerization of this molecule can be used as a signal. Um, in any case, um, these are another factoid to throw out there for you is that these are called vitamins. Not that either one of these has any amino groups, but originally the vitamins, these water soluble vitamins, were classified as vital in some way, and so these are the vital amines or vitamins. So the point being here, though, that uh, there are many roles of lipids beyond fuel oxidation, beyond membrane structure, and even beyond and many roles in signaling. But let's turn away from signaling for a little while and we'll come back to it at the end. I want to talk first about the structure of the membrane. This is a slide that I showed you at the beginning of the semester. Um, there's a lot going on here, but basically giving you the sense that the membrane is a complicated structure. Let's start at the beginning, though. It's composed of a bilayer in which one leaflet, here's the outer leaflet, right, facing the extracellular space, that's composed of, and let's just call these glycerophospholipids, right, these two fatty acid chains pointing in, polar head group pointing out. Likewise, there's an inner leaflet that faces the cytosol that's composed of a similar sort of structure. Fluid, why? So what is this fluid mosaic model? Well, this membrane is held together through a large number of non-covalent interactions. Remember that each fatty acid is only associated, or each membrane lipid is only interacting with its partners through non-covalent interactions, mostly van der Waals interactions. So we can move around laterally and even flip from one leaflet to the other. However, we remember that the membrane is supposed to be a barrier preventing transport of substances across from the extracellular space and into the cytosol. But we mediate transport across the membrane in a variety of ways. Certainly some molecules are soluble in the membrane, and so they can transit simply by diffusion. Otherwise, we control transit through the membrane through integral membrane proteins such as this one, which seems to have a pore, as well as you could imagine maybe transmitting information across the membrane, perhaps as a conformational change in this kind of integral membrane protein. In addition to these integral membrane proteins, however, there are peripheral membrane proteins that are simply associated with the membrane through non-covalent interactions. There are also proteins that are anchored in the membrane. Here's a lipid, carbohydrate, and protein attached to the membrane through that kind of structure. This idea of associating with the membrane through non-covalent interactions with the membrane lipids is going to be important in terms of lipid organization or membrane organization when we get to the end of the discussion. Let's look at eukaryotic membrane composition. Highly variable. Okay, so here this picture is a cartoon of the rat hepatocyte, and depending on what kind of cellular structure we're talking about, here's the plasma membrane. It's a little bit complicated, so let's see. In orange, we're talking about the cholesterol, blue, cardiolipin, which we haven't discussed, um, yellow is other minor lipids, red is sphingolipids, which we have discussed, and then in purple, we've got phosphatidylcholine, and then blue phosphatidylethanolamine. So in one case, a positively charged lipid, in one case, a negatively charged lipid. In any case, we see that what really seems to change a lot here is the relative quantity of cholesterol to phosphatidylcholine. Phosphatidylcholine obviously playing the lion's share in most of these membranes except for the plasma membrane. I won't make too much of this slide except to point out that there is a very wide there are many lipids composing each membrane, and that composition varies from membrane type to membrane type. We'll see not only do these lipid use depend on cellular type or even organelle, but also on where we are in the taxonomy of the organism of life. variable amount of bound or integral proteins, and we'll see that that variable amount of associated protein can in fact depend on the membrane, the lipid composition of that membrane. So we'll see that the membrane composition drives the membrane organization. Ah, right. So another layer of complexity in this story, of course, is that the bilayer composition itself is asymmetric. That is, in the inner leaflet, we have a different organization than we have in the outer leaflet. So they're exposed to different environments, and so you might imagine that that could uh, uh, necessitate a different composition. But the composition is also quite dynamic. Um, the Both surfaces of the membrane have to be able to respond to physiological changes and stimuli. So again, I'm not going to belabor all of these. The main thing is to show you that the two membranes, the two leaflets, are quite different. And the one thing that you might take away from this is that you would say the outer membrane is much less complicated than the inner membrane. Okay. Oh, crying baby.